Leeat Keren. She received her bachelor's in bioinformatics from Tel Aviv University, and this was followed by a master's of science and a PhD in computational biology from the Weizmann Institute of Science. In 2016, she joined the Angelo Lab at Stanford in the Department of Pathology as a Fulbright scholar. She's been the recipient of several other prestigious awards, such as an EMBO fellowship, and she's also a Damon Runyon Cancer Research Fellow as well. Dr. Keren's research focuses on studying the tumor-immune interactions using multiplexed imaging, an exciting new technology that she's going to talk to us about, and specifically its use in understanding the tumor microenvironment of breast cancer. Please welcome Dr. Keren.
Okay, so I would first like to thank Adib and Miriam for inviting me, and I would also like to thank Sasha for this wonderful introduction to why we can't just look at cells, but we also have to look at tissue. And I will tell you today about my work looking at the tumor immune microenvironment in breast cancer using MIBI, multiplex imaging. And I'll start with the second half of this, and this is a technology-oriented conference, and just a brief introduction to why we need multiplexed imaging. And it's because this is how we understand biology. So here we see a T cell interacting with a tumor cell and an antigen-presenting cell. And we understand biology in terms of cells signaling to each other, and this is done in a physical space via a lot of molecules. And so I think historically there have been kind of two approaches to studying tissue biology, the first being imaging. And imaging has been used for decades now, and it's really excellent because it gives us a lot of information. You can see the tissue architecture, you can see the single cell morphology of the cells. However, the quantification is often nonlinear, and of course there's a problem that you can only see very few genes, and so you don't really capture all the molecular complexity of what is happening in these cells. On the other hand, we have all these omics approaches, and we've heard a bunch of them this morning, and these are really terrific because they can measure a lot of genes at the same time and really give a more complete information of what these cells are thinking and what they're doing, but you lose all context for why these cells were expressing these genes. And so today I would like to present to you our efforts in kind of bringing these two worlds closer together, and that is by MIBI. So that's multiplex ion beam imaging, which allows us to do quantification of 40 different proteins in situ. So how does MIBI work? We start with a tissue biopsy, and this could be any clinical tissue biopsy, and we then stain it with a mixture of 40 different antibodies. Only unlike usual immunohistochemistry, where these antibodies are conjugated to fluorophores or to to colors, here these antibodies are conjugated to different metal tags. And so then to detect these antibodies in the tissue, what we can do is measure by secondary ion mass spec. So the way this works is that we basically shoot an ion beam at the sample, and imagine this like big bowling balls hitting the tissue. And every time these bowling balls hit the tissue, they sputter out secondary ions, which can be read by secondary time of flight mass spec. And so for each pixel in the image, what we get is a mass spectrum depicting protein expression in that pixel. And from that, we can then reconstruct the n-dimensional image of the tissue. And here are some examples for how MIBI data looks like. And this is validations in human FFPE tonsil. And just for clarity, I've outlined here the germinal center in green and the interfollicular region in red. And here you can see expression of CD3 and CD20. Importantly, all of the markers that I'm going to show you were acquired in a single experiment. I'm just showing them as individual color overlays. And you can see here CD3 as expected, expressed in the interfollicular zone, CD20 expressed in the germinal center. You can also appreciate that both protein are membranous proteins. Here you can see co-expression of CD3, CD4, and CD8. And so we see a lot of co-expression of CD3 and CD4, CD3 and CD8, but not of CD4s and CD8. And again, these are membranous proteins as expected. We can also detect low abundant antigens such as FOXP3, and FOXP3 as expected is binding in the nucleus of CD4 positive T cells. And here we can also see the characteristic expression of PD-1 on T cells at the edges of the germinal center. Some properties of mass-based imaging and why we like it. The first is that the ability to multiplex 40 channels at once really comes from the low spectral overlap between masses. So this is very similar to CyTOF that we've heard about before. And that means that there are no cyclical stages. The entire mixture of antibodies is prepared at once, everything is stained at once, and everything is spread at once. We also don't require any amplification, so no secondary antibodies because of the system's high sensitivity. And we don't suffer from autofluorescence. And here I'm just showing you a picture of autofluorescence in ovary. And it's a beautiful image, but you can imagine how problematic this is if you want to detect low abundant antigens. And finally, as Sasha stated, it's very important to be compatible with FFPE, which is really the routine way in which clinical samples are stored. And so this allows us to basically go back, and I'm going to show you an example of this, and retroactively analyze close to, let's say, 1 billion archived samples with long-term clinical follow-up. Okay, and so now I'm going to switch gears and show you how we apply this technology to study the tumor immune microenvironment in triple-negative breast cancer. So a few words about triple-negative breast cancer. About 10 to 20%, depending on the classification scheme, of breast cancers are classified as triple-negative. And from the word negative, it's a negative definition, and this is a type of tumor that basically does not respond to targeted therapy, so it does not express ER, PR, or HER2. However, people have seen correlations between prognosis and immune cells, similar to what has been seen in other cancers. And therefore, there have been a lot of clinical trials for immunotherapy. And here I'm showing you, this is probably an old table by now, it's like 2017, where we have here 4 different clinical trials, all of them targeting PD-L1. But what I think is remarkable about this table, other than the fact that all the pharma companies are now targeting PD-L1, is really how patients are stratified into these clinical trials, which I think illustrates how little we know about the tumor immune microenvironment. And so the first clinical trial only includes patients if they express PD-L1 on the tumor cells. The second trial will only include patients if they express PD-L1 on immune cells. And the third and the fourth trials patients are unselected. So this really illustrates that we don't know what immune populations are found in these different patients, which cells express immune checkpoint molecules, and the holy grail is to understand which patients would obviously stand to benefit from immunotherapy. And so what we decided to do was apply MIBI to study immune infiltration in triple-negative breast cancer. We took a tissue microarray with biopsies from 41 different patients and profiled them using MIBI with a panel that interrogated tumor antigens, cell status. A large fraction of the panel was dedicated to looking at different immune cell types. And of course, we also included 4 different proteins for immune regulation: LAG3, PD-1, PD-L1, and IDO, all of them currently under clinical trials for treatment of triple-negative breast cancer. And so this is one of these slides where you show one slide that summarizes a year of work. So this is just to show you that we validated our antibodies, and we think all of them work in tissues. And then after you finish with that big challenge, you have the next big challenge, which is the challenge of data analysis. So a single MIBI experiment, 41 samples, 36 different proteins, generates nearly 1,500 images. So how do you go from 1,500 images to understanding anything about the tumor immune microenvironment? And so we developed a data analysis pipeline, and the first thing that needs to happen in order to really be able to analyze this data and look at all this dimensionality at the same time is to understand where cells are located within this tissue. And this task, as Sasha beautifully explained, is called segmentation. And so I developed a deep learning algorithm to segment the cells out of the tissues, and I'll be happy to elaborate about that later on if people are interested. After we segment the cells from the tissue, then we can basically use all the different algorithms that have been developed for single cell data, so for CyTOF or for single cell RNA sequencing or for CyTECH, and perform clustering of these cells, finding different cell types and different phenotypes. But of course, because this comes from a tissue, you can then overlay this back on the tissue and start asking, how are the cells located, which cells are talking to each other. Okay. So a few of our results, we looked at what is the immune landscape in TNBC. And the first thing we did is we clustered all of our immune cells into 12 different populations. And here you can appreciate that our segmentation and clustering algorithms do quite well, even when the cells are located close to each other on the tissue. as presented here. And when we do this for all of our 41 patients, the first thing that comes out is all these different colors, right? And so there's a lot of heterogeneity between the patients and what immune cells are in the tumors. And so here you can see the general composition. So we find about 25% macrophages, about 40% T cells. But again, this is highly heterogeneous between patients. So some of our patients will have only macrophages in their tumors, whereas other patients will have 75% of the immune cells in their tumors T cells. And so we wanted to try and find some underlying patterns. Why do we see predominantly macrophages in some tumors and predominantly T cells in others? And so the first analysis we did was to take all of our patients and basically rank them by their total immune infiltration. And you can see that this again is highly heterogeneous. So we have patients over there on the top that have hardly any, let's see if I can use this. Patients here at the top that have hardly any immune infiltrate, and we have patients that have a lot of immune infiltrate. And when we do this, one of the things that come out is that the immune composition is highly correlated with the number of immune cells. So you can see that as we go down here, as we have more immune cells in the tumor, we have a higher percentage of T regulatory T cells and a lower percentage of macrophages. And you can see this here visually. And so this made us think that maybe what we're seeing here is some kind of time-dependent progression of the immune infiltration into the tumor. And so to get a better handle of that, we wanted to look at the co-occurrence of different immune populations within the tumor. And so what we did is for each one of our patients, we asked, does it have or does it not have a specific immune population? So for example, patient 24 here on the top has only macrophages, whereas patient 35 here has T cells, has B cells, has NK cells. And so we can then put it into a matrix such as this one, where every row is a patient and every column is a different immune population. And when we started this analysis, we thought, oh, this could look all over the place, right? So some patients could have B cells and NK cells, others could have macrophages and T cells, but it turns out that this is really not the case, okay? So all patients in our cohort that had NK cells also had in their tumors B cells, CD4, CD8, and macrophages. All patients that had B cells also had CD8, CD4, and macrophages, but not necessarily NK cells. And so putting these two results together, what we think we're seeing here is either a time-dependent or maybe context-dependent recruitment of different immune populations into the tumor, whereby specific immune populations either recruit other immune populations or maybe block them from entering into the tumor. Okay, the next thing we wanted to look at was co-expression of immunoregulatory proteins. So do we find that different tumors and different tumor cells use different strategies for immune evasion? Okay, so will I find a situation where, let's say, one tumor cell expresses PD-L1 and another tumor cell expresses IDO, or do I find tumor cells that really try to suppress the immune system, by various mechanisms? And of course, this is important not only on a cellular level, but also on a patient level, right? To determine whether different patients are going to be susceptible to different drugs. So do we find patients which are primarily, for example, positive for PD-1, others which are primarily positive for LAG-3, or patients that are co-positive for both PD-1 and LAG-3 at the same time? And so to cut a very long story short here, the answer is that yes, we definitely see a lot of co-expression, as you can see here, both in the images and in these Venn diagrams. This co-expression is enriched on a cellular level, and that means that a cell that is expressing an immunoregulatory protein is more likely to express another immunoregulatory protein. And this can indicate either that these two proteins have shared signaling, which are controlling expression of both of them, or maybe some kind of positive feedback loop that once a cell becomes immunosuppressive, it kind of turns on that phenotype, and that phenotype is reinforced by various pathways. We also looked at this, of course, at the patient level, and we also find enrichment at the patient level. And so here, for example, you can see that all of the patients that have Tregs in their tumor also express IDO, PD-L1, P1, and LAG-3. And this is, of course, very important for combination therapy because it will suggest that maybe what we do when we give combination therapy is going to help these patients who really isn't going to have into. Okay. The next thing we wanted to look at is we have these tissue architectures, so which cell types are co-localized, and in order to do that, we developed a spatial enrichment analysis, which is basically a statistical way to ask if two different phenotypes in a tissue are co-localized. So let's say if I have red cells and I have green cells, And now I want to ask, are red cells touching green cells more than I would expect just from the sheer number of cells in the tissue and the general tissue architecture? And again, I'll be happy to go into the details of this algorithm for those who are interested later. And when we do this spatial analysis, we find kind of two general characteristics of which cells are sitting next to each other. So what these heat maps are showing us are all of our 36 antigens here in the rows and all of our 36 antigens here in the columns. And if you see dark red, it means that these antigens tend to sit close in the tissue. And if it's green, it means that they tend to sit further away in the tissue. And what we saw is that for some patients, we get this image, which is like patient 16 over here, where you see a lot of dark red and a lot of dark blue. And if you look at who are the antigens, who are the markers that are sitting here in these clusters, we find that these are primarily immune markers and these are tumor markers. Whereas for other patients, for example, patient 2 over here, what we see is that we don't have a lot of very intense colors and we don't have a lot of structure to this matrix. And so this corresponds to two very different tumor immune architectures. The first one here, where you can see immune cells colored in orange and tumor cells colored in blue, where you see these borders between immune pockets and tumor pockets. And the second one, where you see a lot of mixing between tumor cells and immune cells. And so we could then do this for all of our patients and stratify them into one of three phenotypes, what we termed cold. So these are tumors that don't have any immune infiltrate, mixed, where the immune cells are highly mixed with the tumor cells, and compartmentalized, where we find these compartments of tumor cells and immune cells with a very defined border between them. And it's important to note that tumors with similar number of immune cells can differ in their spatial organization. And so we next wanted to put all these results together. So I've shown you that we have different cellular compositions in different tumors, and we have different histologies in different tumors. So can we find a relationship between them? Do we find specific cell types and specific histologies? And here again, for lack of time, I'm just going to posit it. I'm just going to focus on the immunoregulatory proteins. And so one of the things we saw about the immunoregulatory proteins, that these again were heterogeneously expressed between patients. So if we look, for example, at PD-1 positive immune cells, so these are only immune cells that express PD-1, we see that in some patients, for example, patient 14, it's primarily expressed on cytotoxic T cells. In other patients, for example, in patient 35, it's primarily expressed on helper T cells. And you can see this both visually here in the images. And so now we look at the same patients and we sort them by their PD-1 expression, either on CD8 or on CD4. So all these patients have PD-1 expression predominantly on CD8, whereas all these patients have PD-1 expression predominantly on CD4s. And if we now color these patients by their architecture as either mixed or compartmentalized, you see that we see a very good correlation between the two. So these mixed tumors tend to have PD-1 expression on CD8, and these compartmentalized patients tend to have PD-1 expression on CD4. And the same goes when we look at PD-L1 expression on tumor cells versus immune cells. You see this very, very nice correlation between PD-L1 expression on tumor versus immune and the architecture of the tumor, and the same goes for IDO expression. And so we could kind of put all this together into this model where we can connect immune organization, histology, and checkpoint expression, where we find these two different architectures, both of them involving immune cells, the mixed architecture and the compartmentalized architecture, where the mixed architecture will have the PD-1, PD-L1 pathway activated between tumor cells and cytotoxic T cells, whereas the compartmentalized structure will have the same pathway, PD-1, PD-L1, only this time it's between immune cells and T helper cells. And so I was very interested in this compartmentalized phenotype that we found. So what is blocking these immune cells from entering into the tumors? Why are they not mixed? And decided to go a little bit further and explore this tumor immune boundary that is forming between these populations. And so to do that, we developed an algorithm that can automatically identify the tumor immune boundary out of the cell classifications. And you can see this here. So all these blue cells are tumor cells, and all these orange and red cells are immune cells. And we could then, for each one of these cells, we can ask, are they sitting close to the boundary? So for example, in red, we have immune cells close to the boundary, and in orange, immune cells further away from the boundary. Now that we have this tumor immune boundary, we can look at different markers and assess the expression of these markers relative to the boundary. So here, for example, I'm showing you H3K9 acetylation in red and trimethylation in green and trimethylation in red. And you can, I think, even even here, I think you can appreciate the fact that these cells over here close to the boundary are green in the tumor side. And as you go inwards towards the tumor center, they become red, and the same here. And if you don't believe your eyes or mine, we can also quantify that using a rank sum test and establish that really these cells are more acetylated. And of course, this is just for a pair of markers in one patient, but we can do this for many different markers, for all of our markers across all of our compartmentalized patients, and put this into a matrix such as this one. We can then take this matrix and cluster it either by hierarchical clustering or by PCA analysis, which both give us the same result. And we find that these compartmentalized patients basically divide into these three groups. And this last group, this blue group, was the one we found most interesting because this group has a lot of action. You see there's a lot of red and blue happening here. So there's a lot of actioning happening at the tumor immune boundary. So what is happening there? What we can see is that immune cells near the boundary tend to express IDO, one of our immunoregulatory proteins. They also tend to express PD-L1. Interestingly, these tumors are also HLA positive, and we have a lot of speculations about that, and I'll be happy to discuss with people who are interested later. And here's just an example of where we see the exact same organization in a completely different patient. And so just to summarize, we think we can find three kind of main architectures in the tumor immune microenvironment. So we have this cold architecture of patients that don't have a lot of immune infiltrate. If they do, it's mainly macrophages. And we have the mixed and compartmentalized tumors, which have different cellular compositions and different expression patterns of immunoregulatory proteins. Finally, even though this was a very preliminary study, only containing 41 patients, of course, we wanted to assess whether any of these phenotypes were relevant for survival, and matching the literature very nicely, found that indeed they do. So these compartmentalized patients tend to do much better. And I'd like to point out that the x-axis here is years. And I think this really emphasizes the advantage of being able to go to archival clinical samples, right? Because no PhD student or a postdoc will want to wait, 4 to 6 years in order to be able to do this survival analysis. Okay, so with that, I would like to thank the people who did the work. So Mark Mose over here, who was my hands in the lab and did all the experiments. David Van Dalen, who collaborated with us on the cell segmentation. Robert West, the pathologist, who provided all of the samples. And then Sean and Mike. Mike is the guy here with the bad haircut, who developed the MIBI to be. And thank you for listening. Do you think it's intuitive or not? Because we have both. Like people tell me, oh, that's intuitive. And then people say, oh, that's so not intuitive. I'm sorry, I interrupted you.
What is intuitive? Is that mixed with a marker of more
Then more, sorry, macrophages? Oh, more metastases. That's a great question. We didn't have METS information for this tumor. What I will say is that this stratification into compartmentalizing mix is better at stratifying, and again, this is a highly preliminary cohort and very small, but it's better at stratifying patients than the classical stratification if you just immune cells. You take the same cohort and just the number of immune cells. It does separate, it separates responders and non-responders, but not as well as if you add into this the architecture that these immune cells are mixed with. No. So it's not necessarily macrophages. This can also be a lot of T cells. The important thing is that they're highly mixed with the tumor. Let me see if I can find the image. No. It does not segregate. It does not segregate by cell type. See? So here we actually have a lot of lymphocytes. We also have a lot of pockets. They're all mixed together. And here we have this very hard one. Yes. No, these are tumor cells. The tumor cells. And so,
yeah.
yes, that expresses, so the entire tumor expresses DR, and we see a DR gradient where the cells near the border express at the interior. We also see this gradient of acetylation. So that would make sense if you think that acetylation is generally thought of as a marker of active transcription. So maybe these cells at the border, they're more actively driving. I'll say that we also have Ki-67 cells are not necessarily more actively. Ki-67 have higher acetylation. Regarding the HLA-DR expression and here on hand waving, so there are two things that come to mind with HLA. One is of course the pathway. And the second one is that this could be a different strategy for tumors to basically. We know that tumors many times downregulate HIMC class 5. You know, maybe an alternative mechanism. What is happening here is that you kind of bombard the immune system automatically. Yeah.
So that's the end of your talk, but it's so beautiful with the
Yes.
You're saying those two regulate the level.
Yeah, so that's I'm so happy you brought that up because we actually have a different project in we actually have a different project in the lab also looking at field interface, and it seems like a lot of the mechanisms that are looking at this immune interface also happening there. So for example, co-expression of IBO with PD-L1 so yeah, it could definitely be that there are a lot of
My question is about the mass spec contrast using on the tissue. So on the mass spec of the tissue, so you capture the secondary ions on the slice of the tissue, I assume.
Yes.
Do you find under a microscope the best part of the tissue and then you look at it? How does it work?
Yeah, so one of the nice things about maybe that I've skipped due to lack of time, but so you can do one of two approaches. The first one is really go to a microscope, find your interesting regions, and then go and profile these by MIBI. The second is that maybe you can control the amount of ionization, so you can use very high voltages, and so you can basically raster a very large portion, a very thin layer. Okay, and what this allows you to do is that you can actually go and revisit your same tissues. So here, for example, what I'm showing, this is a what we call a large, we call a full tissue scan. It's not a tissue, but this is a 1 centimeter by 1 centimeter breast tissue. And what we did is we scanned this at very low resolution. Okay, so you can't really see all the intracellular features, but you definitely see based on protein expression. And these are expression of 40 different proteins. You can then select, for example, R1 and R2, and then go back into the tissue and rescan these regions at higher magnification. Okay. And you can also use this property of being able to do these rescans in order to obtain 3D information. Yeah.
Back to the question of particulized as you stated, a lot of times in other tools that it's called and you exclude personalized. They're usually the worst outcome.
Yeah.
How do we
Do we have
different compartmentalized?
Yeah, so I think it's actually different terms, and it has to do with the size of the tissue that you're looking at. Because here we've been looking at it as and so they're not immune exclusive. We also have cells in cells, so we have these pockets. It's all about the ratio of what's the boundary of the immune cells and what's the boundary of the tissue. I really think these are coming. And I think that. Oh, okay. And I really think that, I mean, one of the results that we get from these compartmental tumor tend to be better goes together with the fact that we find there is the one highly correlated to work. I think these just the definition of what
tumor cells easier to be killed in the complementary line.
tumor cells easier to.
Killed in the complementary line.
That's an excellent question. So this is again the place where I hand wave and emphasize. So all these patients were not treated by immunotherapy; they were treated by radiation and chemo. And so maybe what happens there is that we don't have any silencing of cytotoxic T cells. So after chemo, we kind of break down this boundary between the tumor cells and the immune cells, and